In this presentation, we'll explore the different levels of mitochondrial DNA testing commonly used in genetic genealogy. We'll discuss HVR1 testing, HVR2 testing, full mitochondrial sequence testing, what each level can tell us, the limitations of each test, and why full sequence testing is generally considered the gold standard today. This presentation is designed for genealogists, DNA enthusiasts, and beginners interested in maternal lineage research. Mitochondrial DNA, or mtDNA, is different from autosomal DNA and Y-DNA. It is passed down through the direct maternal line, mother to daughter to granddaughter. Both males and females inherit mitochondrial DNA from their mother, but only females continue passing it to the next generation. Because mtDNA mutates relatively slowly, it can survive across many generations with few changes, making it useful for studying ancient ancestry and maternal lineage connections. Mitochondrial DNA testing is especially valuable when researching a direct maternal line. Researchers often use mtDNA to compare descendants from the same maternal ancestor, evaluate whether two people share a maternal line, identify maternal haplogroups, and study migration patterns over thousands of years. However, mtDNA represents only a tiny fraction of your total ancestry. A person may have thousands of genealogical ancestors, but mitochondrial DNA follows only one direct maternal pathway. The mitochondrial genome is much smaller than the autosomal genome. It contains approximately 16,569 base pairs arranged in a circular structure. Within the mtDNA molecule are regions that mutate at different rates. The hypervariable regions mutate more rapidly than the coding regions, making them useful for genealogical matching. Older mtDNA tests focused primarily on HVR1 and HVR2. Modern full sequence tests analyze the entire mitochondrial genome. HVR1 was one of the earliest commercially available mitochondrial DNA tests. It examines only a small portion of the mitochondrial genome. Because HVR1 contains relatively limited data, many individuals may share identical HVR1 results, despite having no recent genealogical connection. This means HVR1 matches can sometimes reflect shared ancestry from hundreds or even thousands of years ago. While HVR1 testing helped introduce mtDNA genealogy, it is now considered fairly limited by modern standards. HVR2 testing expanded upon the original HVR1 test by analyzing an additional hypervariable section of the mitochondrial genome. This provided greater detail and improved genealogical resolution. Compared to HVR1 alone, HVR1 plus HVR2 testing reduces the number of identical matches and allows for more refined maternal lineage analysis. However, even with HVR2 added, large portions of the mitochondrial genome remain untested. Because of this, many genealogists eventually move toward full sequence testing. Full mitochondrial sequence testing, often called FMS, analyses the entire mitochondrial genome. This includes both hypervariable regions and the coding region. Because the full genome is examined, FMS provides far more precise matching, better haplogroup classification, and much deeper maternal lineage analysis. Today, full sequence testing is generally considered the preferred option for serious mtDNA genealogy research. In mitochondrial DNA testing, genetic distance refers to the number of differences between two mitochondrial DNA results. For example, genetic distance 0 equals an exact match. Genetic distance 1 equals 1 mutation difference. However, mitochondrial DNA mutates slowly. This means even an exact full sequence match may still represent a shared maternal ancestor from many generations in the past. Unlike autosomal DNA, mtDNA alone usually cannot pinpoint a recent relationship without traditional genealogical documentation. One major advantage of full sequence testing is improved haplogroup refinement. Older tests may only identify a broad maternal branch. Full sequence testing can often identify much smaller downstream subclades representing more recent maternal population branches. This helps researchers better understand ancient migrations, population history, and relationships between maternal lines. This chart summarizes the major differences between the three testing levels. As we move from HVR1 toward full sequence testing, the amount of usable genealogical information increases significantly. Today, most advanced mitochondrial DNA research relies on full sequence testing because of its improved accuracy and resolution. While mtDNA is extremely valuable, it also has limitations. A mitochondrial haplogroup alone cannot prove tribal enrollment, ethnicity, or a specific historical identity. Responsible genetic genealogy combines DNA evidence, historical documentation, traditional records and careful analysis. DNA is one tool among many. As mtDNA testing technology improved, genealogists moved increasingly toward full sequence testing because of its superior detail and reliability. While HVR1 and HVR2 played important historical roles in the development of genetic genealogy, full mitochondrial sequence testing now provides the best opportunity for meaningful maternal lineage analysis. Thank you so much for joining me for this presentation on mitochondrial DNA testing levels and maternal lineage research. In future videos and presentations, we'll continue exploring genetic genealogy, chromosome mapping, haplogroups, autosomal DNA, Native American genealogy, population history, and advanced DNA analysis techniques. If you enjoyed this presentation, please like, subscribe, follow, and share.